Hi, everyone. Thank you for joining us. We are going to use this time together today to go through different examples of non-Mendelian inheritance patterns. So yesterday's notes assignment will be very helpful to you to have available as I talk you through these examples of non-Mendelian inheritance. I'm going to get rid of those pictures. All right, so I do not see anyone if you're showing your video right now because I minimized it so that I can see and focus on the material on my screen. You should at this point see my PowerPoint notes that I am about to start going through. This isn't my way. Get this out of here. Okay, so what I also want to point out to you is all the notes that are linked on my website. If you see links on the bottom, like this one, for example, they're live links on my website. So you can click and watch those videos. That's something I would click and show you if we were having an actual lesson in our classroom. So it would benefit you to click and watch those videos. So if you didn't do that, if you skipped it just to get the note sheet done, just try to keep in mind they're there for you to help you learning. So if you have time, go back and actually watch the videos, even though it doesn't specifically make you do anything. It's just there to help you. Okay, so oh, someone's trying to join. Sit tight. Latecomer. Let me get back to it. No one's trying to join. Oh, maybe somebody left. I got a ding dong sound. That happens when somebody leaves and when somebody joins. Oh. Nope. Okay.
You need to eat, so are you okay?
Okay, so I'm going to remind you again to please mute your microphone unless you have a question. And we're going to get started now. Thank you for your patience. Okay, so to remind you about Mendelian inheritance, because we're about to compare non-Mendelian inheritance. So Mendel worked with pea plants. They had only 2 forms of each gene. We call those alleles. And so it was either tall or short, dominant or recessive. So you still have to understand that concept of dominant versus recessive, homozygous versus heterozygous, genotype versus phenotype. All those terms are still applicable. We still got to understand them. We still got to know Mendel's basic principles and laws. The idea that one factor or one allele, now that we call it allele, he called it factors, can dominate over the other. We call it dominant recessive. All that's still important, but now we're going to kind of go beyond what he talked about. So his law of dominance, his law of segregation, his law of independent assortment still applies to some degree, but we're going to see mainly how the law of dominance is getting kind of thrown out the window for these examples of inheritance. So when we talk about non-Mendelian inheritance, we have inheritance patterns such as incomplete dominance versus codominance. Now this video that I do have linked in my notes directly, I'm not going to play it for you now while we are, I did click on it, but just to show you, while we're talking to each other, I don't want to play it for you now, but you could go back in and click on it yourself. All right, this is an advertisement. I'm not going to waste our time going through the ad, but it's going to be these little color-coded little puffy balls, like blue balls and yellow balls and green balls, and it's going to help you see visually how these 2 inheritance patterns differ. Incomplete dominance versus codominance. So they are the first 2 non-Mendelian inheritance patterns we're going to talk about. Then we're going to talk about multiple alleles, which can also be included with codominance in some examples like blood typing, and polygenic inheritance. So in reality, most traits, especially human traits, don't follow Mendel's rules. I told you that big disclaimer the first day that we were talking about Mendel. I told you about the experience I had when the student in the class I was sitting in and observing realized that her parents both had blue eyes, but she had brown eyes, and realized that that couldn't be possibly true according to just Mendel's basic principles. And the teacher embarrassed her, wrote mailman on the board. But so he could have pointed out and reminded her that, in fact, most human traits don't work that simply. So we're going to talk about examples of non-Mendelian inheritance. The first one is incomplete dominance. So this is just when you have a heterozygous genotype, so meaning one big letter, 2 different alleles, you're going to see a blending of the original 2. So you're not going to see either of the original 2 traits. You're going to see a blending. So for example, white and red make pink. So you can have a white parent plant and a red parent plant make all pink baby plants. So it's a completely new phenotype that shows up different from the parent's phenotypes. What this means is that neither of those traits was dominant. And so we end up seeing a blended phenotype of both those original 2 parent traits. This example of a flower, there's Japanese 4 o'clock flowers, there's snapdragon flowers. A lot of fossil flowers work this way in determining petal color. It's an incomplete form of dominance. So what we do for the notation here is where it's going to change a little bit versus just plain old Mendelian inheritance. We are used to using the same letter and using a capital and a lowercase. We can't do that now, though. We're going to use this. We're going to use a letter for each of the two different traits. So, for example, red flower, we're going to use an R for red. White flower, we're going to use a W for white. We're doing this to distinguish and show this is not regular Mendelian inheritance. So our notation changes. Okay, that's an important thing to notice. So our key is going to be capital R for red, capital W for white. So in our example, we had a red parent plant. So if it's a red parent plant, it's got to have two R's. And we had a white parent plant, so it's got to have two W's. And if you cross a red parent with a white parent, you end up with all the offspring inheriting an R and a W. And none of them are going to appear red, none of them are going to appear white, because instead what this combination produces is a blend of those two phenotypes. So if you take some red paint and some white paint and you mix it up, you see it turns pink. So the genotype, phenotype of the offspring is going to be 100% RW, the heterozygous, and 100% pink. So none of the offspring show either of the parents' phenotypes. This is called incomplete dominance. Okay. I'm going to pause and just quickly say, unmute yourself if you have a question about incomplete dominance. Blend. The key word here is blending. You see a blend of both of the traits. This is going to be a little bit different from codominance, and this is just another practice problem. Incomplete dominance, again, we're using an R and a W to show red versus white. Now, if we want to cross pink flowers, pink parent has RW, and a pink parent has RW. Bless you. And then when you get those offspring combinations still, then now you see the original red genotype coming back and the original white genotype and phenotype coming back. So now we will end up seeing 1 out of the 4, or 25%, have the two R's, 2 out of the 4 have RW, and 1 out of 4 has 2 W's, which would of course produce white. So we have a 25, 50, 25% ratio of those inheritance, of those genotypes and phenotypes. Okay. Now, when we're doing codominance, that's the second inheritance pattern we're going to talk about today. Instead of seeing a blending, I'm just going to ask if you mute your microphone if you don't have a question. Some background noise, please. Thank you. Like papers or shuffling or something, just mute your microphone. Thank you. Okay, so now codominant, like if you're on a team and you have co-captains, or if you're in a club and you have co-presidents, or if you're in a classroom and there's 2 teachers, you have co-teachers, both teachers, both presidents, both club captains are both in charge. They're both together going to show up and do everything. So codominance means both alleles are dominant. Neither one is backing down. There's no blending happening here. So in codominance, we see both traits actually show up. So this is illustrated in cattle. So we have red cattle and white cattle, and they can be crossed and produce what we call roan cattle. I need somebody to mute their microphone, please. Thank you. Julia, I think it's you.
Toss, I put out food, maybe try to get them to eat it.
They eat it yet?
Julia?
Not even a treat.
All right, I'll do it.
So they really could use some food,
I woke up at 9, like a minute
and they could really use some walk.
A minute later, my English teacher started her live, but
All right, there we go, took care of it. Okay, back to codominance. So both traits are going to show up. Both the red and the white are going to show up in this type of inheritance pattern because both are dominant. So we call it codominance. There we go. So we call these cattle roan. Roan is just when you see the spots of both of the phenotypes showing up. So both white and red hairs get expressed in these roan colored cattle. Now you're going to notice, oh, we have red and white. So that's going to be R and W again. But we have to show that codominance is different from incomplete dominance. So where we had previously for incomplete dominance just used capital R, capital W, that's the notation for incomplete dominance. We have the same alleles here for codominance. We have to distinguish that some way. So to distinguish that, we're going to use a C in our genotypes, a capital C, and then a superscript with the allele. So I'm going to show you that now. When we practice codominance, we're going to cross a red cow with a white cow, like we just crossed a red flower with a white flower. We're not going to get pink cows, though. We're going to get cows that show both red and white spots. So our key has to be different. Our red cow allele is going to be capital C with a superscript R, okay, just like this, like an exponent R. And the white allele will be capital C with the capital W. So we're still using the R's and the W's, We're just showing that this is different inheritance pattern by throwing this big C in here, C for codominance. So we're just showing it's codominant inheritance rather than incomplete dominant inheritance. So that way, when we cross a white cow with a red cow, we're going to use CW for the white cow, CR for the red cow. Okay, and when we write it in, we're going to see we have that 100% heterozygous genotype in all the offspring, but we have to know how to interpret this. And that's why we make our notation different, so we can interpret it different. So we know that these cows are not pink. We're not seeing a blend. That's incomplete dominance. Now we're seeing both alleles physically show up. We're going to see patches of red and patches of white, and we call that roan when we're talking about cattle color. So both of them do show up. And that's how you have to see the notation differently so that you remember to interpret the phenotypes differently. Okay, so don't confuse incomplete and codominance. Next up, one more practice problem. So if we take those roan cattle, they have the heterozygous genotype. They're going to be the CRCW and a CRCW. And when we cross them, it looks very similar in terms of percentages to when we did the incomplete dominant cross with the flowers. So now we're going to get 25, 50, and 25 again. But now again, we see the original red coat and white coat come back. Okay, the heterozygous is where it's different. The interpretation of the heterozygous is different because both of the traits show up. Okay, and ratio is 1 to 2 to 1 still. So that's incomplete dominance versus codominance. This is just an illustration with pictures of flowers to show how the phenotype would come out differently if you had incomplete dominance happening versus codominance happening. So these two flowers at the top, let's say they're my parents. If a red parent and a white parent are crossed and you get this situation on the left, you get a blend of pink, that you have to realize is going to be an inheritance pattern that is incomplete dominance. If you get no blend, if you get stripes on the right here, you get both the red and the white showing up, then you're getting codominance showing up, codominant inheritance pattern. All right. Do the same thing again, just clicking through. The next item on the list to talk about is an example of multiple alleles. And so I often would start the lesson by talking about blood type, who's donated blood at our blood drives before, and then you get a blood card in the mail, you know your blood type. I like to make the joke that my blood type is A positive, just like my greens. So a lot of people are actually A positive, if you see here by this breakdown, about 34% of our population. But what does that even mean? So if you think back to your living environment days, quick little recap, this is a way of typing which antigens your cells are used to seeing. So my cells are used to seeing the A antigen. So A is normal for me. B is not normal for me. And so this is important when we're talking about transfusions and organ donations, but the determining factor of this is genetic. And so that's why it comes up in this course. You can get A blood because you get an allele from your parents that is the A allele, and that is the allele for producing the A antigen, and so we call you type A blood type. My husband is type O blood type. That O is more like a 0. It means you don't have any A antigens and you don't have any B antigens. And so if you happen to get introduced to A or B antigens, like if there was a transfusion that didn't match him, his body would attack. His body would say, nope, this doesn't belong here, and I would attack that blood or attack that organ. So O is like a 0, and you would have to get the O alleles from your parents. And in fact, you'd have to get 2 because O is recessive. So this blood type scenario is a way of combining our understanding of dominant recessive, a way of combining our understanding of codominance, because 2 of these types are codominant to each other, and an example of multiple alleles, because there's more than 2 possible alleles you can get for determining your blood type, and we call that multiple alleles. So that's why we introduce blood typing here. So multiple alleles, I asked you to jot down some information on your notes chart, is when there's multiple or more than 2 allele pairs for a gene. You still can only inherit 2 because your dad gives you 1 and your mom gives you 1. But in the population, there are more than 2 possibilities. Okay? That's what a multiple allele trait is. So in blood groups, we actually have 4 different blood types based on 3 different allele combinations, not combinations, but 3 different alleles that you can get various combinations of. So you can be type A, you can be type B, you can be type AB, or you can be type O. And those blood types are produced by different allele combinations. And there's a good Amoeba Sisters video on this that would be helpful for you to go ahead and watch. Tomorrow, I'm going to probably assign you a few ed puzzles to help review these different inheritance patterns. One of them might be that one. So some facts you got to know about blood type and how they apply with genetics is that A and B are what we call codominant. Remember, that means neither of them are backing down. They're both going to show up if they are present. So if you have an A allele in your genes, you're going to make the A antigen. If you have a B allele in your genes, you're going to make the B antigen, and therefore you will make both A and B antigens, and so you will be blood type AB. So A and B are codominant to each other, and O is a recessive allele. That's something you got to kind of have, you got to memorize, especially if you're going to do the shorthand notation I'm about to teach you. There is a more formal notation where that does illustrate O as recessive, and that is technically more correct, but I see it both ways in questions. So you got to know these facts. A and B are codominant to each other. O is recessive to both A and B. So the only way for O blood type to show up, like in my husband, is for him to have 2 O alleles and no A and no B alleles. That's how O blood type shows up. All right, so the phenotypes and genotypes you have to recognize the notation of. So type A blood, like me, I could be type A where I have 2 dominant alleles, and we use this capital I here as sort of like a placeholder, like we used the capital C before for codominance. The I actually stands for immunoglobulin as a blood protein. So this is like saying I have immunoglobulin A and immunoglobulin A, and I have 2 of them, so I'm homozygous dominant for the A allele for blood typing. Or I could be heterozygous for type A, the second option here, where I have only 1 dominant allele of A, and then my second allele, we write a lowercase letter I to represent the O allele. So lowercase I is the recessive form of this gene. I don't have an A allele, I don't have a B allele here. This lowercase I is just an O allele, excuse me. This shorthand notation, which I actually prefer, is just faster to write, is written this way. So what you see here on the left is the same thing as what you see here on the right. It's just writing it in a different notation. Oh, too fast. So homozygous dominant A blood can have capital IA, or shorthand it, just writing 2 capital A's. When you do the shorthand notation, though, you have to remember that O is recessive, because when you write the heterozygous form, you're writing just an A allele and an O allele. You have to know that's heterozygous, but you have to know O is recessive to A, and so this person's going to show up with type A blood that O is masked, like any other recessive allele. The more formal notation for heterozygous is this one here. That is technically more accurate in determining and showing that O is recessive, because we're showing that recessive lowercase I allele instead. So for type B blood, it's going to be pretty much the same options, except you're going to have B's in the places of where the A's are. So this is a person with type B blood that is homozygous dominant. This is a person with type B blood that's heterozygous. Or you can write it in the shorthand notation. Homozygous dominant B, heterozygous B. Okay. Someone with type AB blood is only one way to do it. Well, there's the two notations, but only one combination. You have to have both an A allele and a B allele, and remember they are codominant to each other, so they're both going to be expressed and show up, and you'll make both antigens. So the shorthand notation, you just write AB. And type O blood, if you're using the formal notation, you're using 2 lowercase i's for type O blood. I know it's weird sounding, like why am I using i's for O? That's just because it's recessive, and we're showing that you need 2 of the recessive alleles for it to show up in the phenotype. The shorthanded way is writing 2 O's. So I'd like you to practice this both ways. I believe I gave you space for doing it both ways in your blood typing examples. I don't have them all written out here though. So try them, and then what I can do is I think I have a written out answer key. Like in my binder, I could take a picture of that and send that to you guys later. The last example of inheritance that's non-Mendelian is called polygenic inheritance. And this is pretty much everything that you can think of that makes up human traits. So most of our traits are coded for by more than 1 gene. So most of our traits can't do a simple little 4-box Punnett square because there's more than 1 gene involved in determining those traits. So we call this polygenic inheritance. Poly literally translates to many, so it means many genes, many gene inheritance. There's an Amoeba Sisters video clip here. I suggest that you go and watch. It's available through my notes on my website. So this is just any characteristic of a human that has a variation along a continuum. So it's not just tall and short. Like in pea plants, you can be tall or short and that's it. But in humans, we can be anywhere in between a range of tall and short. And so this range is coded for by many, many genes. So there's many genes that code for our height. There's many genes that code for our skin color. our eye color. Most human traits are going to be polygenic inheritance. There's several separate genes that are going to help code for them. So skin color, eye color, height, etc. So this chart, this graph is showing you this range of possibilities that are going to be possible with a polygenic trait. Height is one example, skin color is another example, eye color is another example. In fact, most human traits are polygenic. I love this example. I think I've shown you these lovely twins before. These are twin sisters. They are fraternal twin sisters. They really help demonstrate polygenic inheritance of skin pattern well because they came from the same parents' combination or possible combination of gametes. So if you wanted to do a Punnett square, it's really not a Punnett square anymore because it's enormous, for skin color, we think we have found there are at least 8 different possible genes that influence skin color. And so if you were to kind of like code them into number form and how many of them you have versus how many of them you don't have. This is like a simplified version of trying to show how ranging skin color is. Those 2 parents could produce the range from this very, very light skin tone here in the top left box to this very dark skin tone here in the bottom right box. Okay, so skin color is one of the many human traits that are polygenic. There are a couple more terms on that note sheet that I did want you to make sense out of, and this is going to be helpful for when we get into doing an activity later on, probably after the April no more break time. We're going to do an online gaming activity that practices different types of inheritance patterns. It's fun. It's called pigeonetics. So this example, this term you need to know is it's called pleiotropy. So this is when a gene affects more than one phenotypic trait. So examples that I had you look at is the mutation that causes a collagen problem. It's going to then affect other phenotypic problems. So that one gene can have multiple problems phenotypically being expressed. It works with sickle cell anemia too. Having sickle cell anemia is going to cause you to have malformed hemoglobin protein on your blood cells, but that's going to also phenotypically impact other parts of the body. And so this is pleiotropic in that sense. So one gene affects more than one phenotype trait. So here's the gene for sickle cell anemia. If you have that mutation for it, it's going to impact not only your red blood cell proteins, but it's going to impact blindness, liver failure, heart attacks, increased risk, etc. And then the last term, I think this was the last term I put on the list, is a term called epistasis. And you actually saw this already in the very first thing I had you do in this class when we made our pigeons, and I gave you some chromosomes and you had to follow the steps and create your own pigeon and color your pigeon. If you remember, you had one possibility, or some of you had the possibility where a trait you had the genotype for, but there was another trait that kind of crossed it off and said, oh, if you have this one, then this one is eliminated. This is an idea called epistasis. So the expression of some genes can impact the expression of other genes. So you might have a genotype that codes for making a certain wing pattern in pigeons, but if you have a genotype for something else, you're going to see that it kind of crosses off and negates that wing pattern. An example in humans is the gene for albinism. So albinism is a genetic skin disorder. That person doesn't make skin pigment. They're very, very pasty white skin and white hair and red eyes because they don't make any of that melanin pigment. So this is going to be an epistatic controlled situation. So they might have skin color genes from their parents that code for making a certain skin color, but since they have this albinism gene, that one kind of epistatically controls and eliminates the use of the skin color genes. Okay, so epistasis. A visual example you can see is if you look at Labradors, the black lab, chocolate lab, and yellow lab are produced via epistatic control. So there's one gene that is going to negate and turn off whether you have you could have the genes for black, but if you have this other gene that has epistatic control, you don't express the gene for black and you show up yellow. So that's how these dogs work. Epistasis. All right, that's that. And then we're not going to go into pedigrees because that's a whole nother thing. Okay, so that is my lesson on non-Mendelian inheritance. Do you have any questions? I'm going to go back to seeing you because if you raise your hand or anything or look like you have a question, there we go. Make this bigger. All right, so just unmute yourself if you have a question. I know I went through that kind of quick. I normally would teach that in multiple lessons in the classroom, but you already had the notes done, so I thought we could go through it pretty quickly. So unmute yourself if you have a question, or if you want to come on video and raise your hand, you're welcome to do that as well. Everybody stay muted. So I'm thinking that means you're good or you tuned out, one or the other. Hopefully just you're good.
I have a question, Ms. Miranda.
Yes, hi Sal, what's up?
So if we do the other assignments, do we have to log into the Zooms?
So my goal for the Zoom is to kind of like teach you whatever you need me to teach you if the material is not doing it for you. Some people really need that direct instruction more than others. The Zooms are still optional, they're not mandatory, and I'm going to record them and post them on my YouTube channel if you want to be able to go back and look at them later if you can't watch them live. So it's up to
Okay,
you.
thanks.
You're welcome. Any other questions? Okay. All right, I see some of you bowing out. If you don't have any other questions, then I am going to end the recording. If you decide that you want to hang back and ask me anything, you can hang back. I'll stay here till everybody's done.